Hi, I think we have everyone or almost everyone now. Um, Hi, everyone. We will get started in just a second. We're just doing some last minute tech stuff, so please hold on. Okay, can I get a test of the interpreter just so we can make sure that we can see you and that the audience can see you?
It's not giving me access to start my video. Yeah, it's saying unable to start.
Okay. We are having some technical difficulties, so please bear with us. No, don't change role. Shoot. Can all the panelists please turn on your video for a second? We're just trying to make sure that we can see everyone. Or are you all also getting the cannot show video?
Same message of cannot show.
Yeah,
video.
same message.
Me too.
Yeah, I cannot start your video because host has stopped it.
Same message.
Yeah, same.
Okay.
Okay,
Give me
got
a
it.
second. Using the view. Yeah. Meeting. Rashonda, do you have video now? Okay, I got it. Okay. Okay, so Lavita, you should also have video now. learn play sign? Yes, okay, I got it figured out. Apologies, everyone, we were having some technical difficulties, but everything seems to be in order. So basically what's gonna have to happen, Rashonda, is that as each person goes, you have to make them a co-host, and then their video will be able to go through. Okay, all right, we're ready to start now. Apologies for the delay, everyone. Thank you so much for coming to the Early Career Research Symposium. We are really excited today to get to see some of these wonderful Black microbiologists share their research. Hopefully we'll have a lot of good questions at the end, and hopefully you enjoy what we have for you today. So you can go ahead, Rashonda, and I'm going to turn my video off. You're muted.
Okay, I think I accidentally demoted this first speaker.
Yeah, I got it. I got it.
Okay, okay.
Daisy has been upgraded. You're good. You're good.
Okay, so for our first speaker, we have Adais Gubufor, and she is from the Institute of Biological Sciences at the University of Malaya Laboratory in Malaysia. And she's going to talk to us about isolation and characterization of foodborne microbes from leftover food. So I'm going to turn it over to Adais.
Okay. Can you see my screen?
Yes.
Okay, so welcome again, everyone, to Black in Microbiology Week and to the Symposium section. As I've already been introduced, so I'll be taking the lead of my research, and I will just quickly go in. So my research is based on the fact that I'm trying to solve some public health issues, especially from leftover food. Sometimes they are being ignored. So I'm trying to create this awareness of these microbes that still can be found in our leftover food. And for my content, I will be taking you through my research background, the problem statement, and the objective, the research methodology, a few of the results and discussions and the conclusions of my research. So for my research background, I think maybe not everyone is from the microbiology field, so I will be a little bit, break it into a little bit lower terms so everyone can understand. As we all know, most of us or almost everyone consume leftover food, and they are being considered as high risk food because they have the high tendency to be infected by pathogens and microbes because they are being eaten late hours or days after they are being consumed. And some of these foodborne illnesses or microbes such as the Campylobacter species, the E. coli, the Salmonella, the Shigella species, and also the Bacillus species. And one of the things that we should know about foodborne microbes is that it can either be a virus or it can either be a mold or fungi. But those that cause the food poisoning or the food illnesses, especially for diarrhea diseases, are caused by the bacteria. So this is going to be what of our study. So in Malaysia, one of the cases that occurred was that 32 students actually were hospitalized after they consumed a food that was supplied to the school around 11 p.m. So these students actually didn't consume that food immediately. So some of them, when interviewed or asked what could have been the situation, they said they had consumed that food at 11 a.m. and some of them had consumed that food at 5 a.m. So we want to assume that since the food was not consumed immediately, and this food is made up of beef and chicken, and it's called nasi Arab. And so we want to assume that probably the food would have been infected with pathogens as it has become a leftover food. So the basis of this study was first trying to understand what type of foodborne microbes are actually found in our leftover food, and also at what time do these microbes infect our food. So we try to study these foodborne microbes based on the food temperature danger zone, which is between the 5 degrees and the 60 degrees. So for my study, I focused on two major temperatures, which is at the 4 degrees, which is the freezing temperature, and at the 25 degrees, which is the room temperature. And one thing we should also know about microbes is that they replicate very fast and they produce toxins based on the conditions, on the environments that they find themselves. So two major objectives was first to know what type of foodborne microbes are in our leftover food, and also to know at what particular time they are infecting the food. So the research methodology that was used here was first the spread plate method after the samples were made into a liquid form using the serial dilution method. And we performed the spread plates method, and then we did the streaking method, and we went from the gram staining. The gram staining helped us to understand what type of bacteria they are based on their morphology, if it was gram positive or gram negative. And then the study, we further collected those pure cultures and pure colonies and performed a selective media test in order to differentiate what type of microbe were actually growing from those colonies, those pure colonies. And we further did a biochemical test. If it was positive or negative, we were able to record our findings and further molecular characterizations using the PCR test. And the result that we received was at the 25 degrees, which is the room temperature. We discovered that the certain time that you keep your food, even at 0 degrees when the food was freshly purchased, which was the meat and the chicken, we discovered that there were already microbes already growing there. And so we tried to study it at hours and by day. So at 24 hours, we discovered the highest growth of microbes on the leftover food at the room temperature. So it already shows us, that gives a signal that even with the control of these food samples, there were already bacteria growing there. So even the more time you keep your food at the room temperature, the higher the microbes were growing. And also at the freezing temperature, we discovered at day 6 of the food, if in the freezer, there was already microbes growing. Even though your food was in the refrigerator, you could say, okay, yeah, it's in the refrigerator, it's fine, nothing is going to happen, there is no pathogens growing there. Like I said, microbes can grow at any temperature or any conditions that they find themselves. So we provided a few of justifications for our research. And furthermore, from the pure cultures of the food samples, we were able to grow them on a nutrient alga and try to understand what type of colonies were actually growing. And further, the results from the ground staining showed that we discovered 36 strains of bacteria were actually growing from the 2 food leftover samples. And 22 of them were coming as gram negative, while 14 of them were coming as gram positive. And we further studied what was their morphology, and it was further classified. So for the results that we had on the selective media, we used different medias that actually could grow these certain types of foodborne microbes. So we discovered some of them had no growth. For those that had no growth, it was classified that that bacteria or maybe that colony or microbe was not a foodborne microbe. Maybe there could have been there were actually other types of microbes growing, but we didn't classify them as a foodborne microbes. And we discovered Staphylococcus species were actually growing in our colonies. And at the time of the research was taking place. And furthermore, we further classified it a bit based on the results that we had on our selective media plates, and if they were showing the certain color changes. So if it was changing from red to yellow, we identified it as Staphylococcus was growing there, or if they were changing from just pure colonies to maybe changing colors to white or black, we identified them as Campylobacters or other types of microbes were growing there. So furthermore, for our classifications, we discovered that on a scale of 100% for the 36 strains of bacteria that we found, 36% of the bacteria that was found on the leftover food was coming from Staphylococcus. And this was justified to the fact that in our chicken and meat products, it doesn't matter if they were cooked or uncooked, naturally from the farm or wherever the produce has been gotten from, they already contain Staphylococcus species. So as individuals, like if we purchase this type of products like chicken or beef, whether cooked or uncooked, we should take extra care in making sure that not only it is being cooked properly, but it is being protected even after the food has been cooked, and it should be properly stored. And this was occurring at the 25 degrees, which is the room temperature. And then you could even wonder that, okay, this is a room temperature. Yeah, I accept the fact that, okay, microbes can multiply very fast. But how could these same microbes still be growing at the refrigerator temperature at 4 degrees? And we still discovered that these same Staphylococcus species are actually still growing at a higher rate of 25%, and we discovered other bacteria growing at 50% or 36%, but they could be coming from the utensils that were used in preparing the food, or the water that was used in preparing the food, or even the individual that prepared the food. So this was the last test we used to further classify the type of bacteria that we had. So at the Manito alga selective media plates, we discovered that it was Staphylococcus. And at the Palcom alga plate, we discovered that it was Listeria species. And at the Campylobacter alga plate, we discovered that it was Campylobacter species that were growing, and Proteus species of microbes were also growing on the plates. So for the conclusion of my research, I would like to highlight a few things. Of course, I already did, that we discovered like 4 major bacteria growing from a leftover food. And these bacteria that grew were based on the different time frame and the temperature conditions that they were subjected to. And we discovered that more bacteria were found at 25 degrees compared to 4 degrees. So we can probably say the food you keep at room temperature has higher tendency to be contaminated by these micropathogens than those you keep at the refrigerator temperature. But it doesn't necessarily mean that those at the refrigerator temperature are safe. So we still need to control the number of days that we keep our food. And also the research concluded that although the bacteria were isolated from the food, we discovered other bacteria, and we just focused on just those foodborne microbes for our research. And just a few precautions that while we take care of our leftover foods, as individuals, or maybe those that are working in the restaurant that handles the leftover foods that have been that could not be sold at that day, or maybe you are preparing for your family at home, or maybe you have anniversary, or the leftovers that come from it. We should also be careful to know that there are people that are actually prone to food poisonings, and these are like the elderly people aged 65 and above, younger children, younger than age of 5, those that are on medications, pregnant women. So they are the ones that should take even extra precautions while handling their leftover foods. And in order for us to prevent food poisoning or food contamination, we should try to make sure that we wash our hands, the utensils, the kitchen surfaces that are being used to cook the food are being properly protected, because as you can see from the research, we still had over 50% of microbes still growing out from the food. So this could have come from the utensils or maybe the individuals handling the food. And make sure that we separate those, keep the fresh produce away from the raw food, because these microbes can actually interact. And also when you're keeping the food in your refrigerator, it should be within 2 hours before you keep them in the refrigerator, so you avoid keeping your food at the room temperature. And furthermore, if you feel symptoms of diarrhea or more than 3 days, you should already know that you are having symptoms of food poisoning, and it would be good that you consult with your doctor, because even though we preheat our food or properly cook them to the temperature, sometimes these microbes have the tendency to still replicate and produce their toxins. So extra care, I would like to create the awareness that extra care should be taken even while consuming your leftover food. And yeah, this is the end of my presentation. Thank you.
All right, thank you. So we're gonna allow 1 minute for questions. If there's any questions from the audience, you can put it into the chat now. I'll start with a quick question. About so I saw that there was more foodborne illnesses, more foodborne illness related microbes associated with the chicken and not the meat. Is that correct?
Yeah.
And if that is the case, why do you think the meat didn't have as much? Is it just how it was handled, or can you speak more to that?
Yeah, like I explained, like these foodborne microbes, they they replicate based on the conditions and the temperatures that they find themselves. So even the fact that the food was kept at the same temperature, different types of microbes were growing. So maybe at the type of microbe that was growing at the chicken might be different from that was growing with the meat. So I think that's why at that time we had more microbes growing from the chicken. And what we did was, despite the fact that more microbes was on the chicken, we decided to only isolate those that are foodborne microbes, and which was also found in the meat. So other types of bacteria that probably made it higher could not be coming from the food. Probably they came from other sources that associated with the food. So yeah, I think that was one of the reasons why, and we were able to find a few studies to support that, the higher prevalence of the bacteria on the chicken.
All right, well, thank you for that. So now we're gonna turn it over to, we're gonna turn it over to Aya Donovan Banfield from the University of Liverpool in the UK. And she's gonna talk to us about in vivo viral population dynamics of SARS-CoV-2 and patients enrolled in the Agile CS2 EIDD 2801 trial. in the UK. All right. So Aya, you can get ready to start.
Okay, thank you. Can you see my screen and my pointer?
I can see this. Yes, yes.
Yeah, okay. Oop. I'm just trying to hide the panel. Yeah, so Shonda said, I'm just gonna give a quick presentation on the work I've been doing since the start of my PhD earlier this year. So I'll go straight in. So during the SARS-CoV-2 pandemic, there's been a push to, as well as develop vaccines, to develop antiviral treatments. Some of these antiviral treatments have been repurposed from previous drugs, and some have been newly created in the time since early 2020. And one of them, one of the repurposed drugs is remdesivir, and it's had varying successes in trials, but its route of administration is via IV drip, which is not convenient for treating outpatients, which the majority of COVID patients or people who are infected with COVID are. So one of the other ribonucleoside analogs is this EID 1931, which was developed at the Emory Institute for Drug Discovery. In 2019, they developed a more orally bioactive version, this prodrug, which is EID 2801 or molnupiravir. And because it can be administered via an oral pill, it can be used as a rapid treatment after your positive test, or it can be used as a prophylaxis if you're going to be in high risk environments. And the method of action of this drug is that it induces aerocatastrophe in the virus so that it cannot replicate and cannot sustain infection in its host. And the aim of my study is to look at samples taken from this Agile Coronavirus Drug Testing Initiative in the UK and look for hallmarks of the mechanism of action to validate it, including effectiveness against the different variants that have arisen in the UK. So during the trial, of which there are 120 patients currently enrolled, each patient has a nasal swab taken on the first day of their treatment or placebo, and sequentially throughout for 28 days thereafter. The RNA is extracted from those samples and processed by qPCR for quantification and confirmation. I receive a small aliquot of the RNA from these extractions to sequence and look at the genomes. So what I do with the RNA is I reverse transcribe it and I PCR amplify to create an amplicon library for each sample. The amplicon library is made according to the Arctic Network amplicon scheme, which consists of 99 amplicons that go across the whole length of the genome in a tiled fashion. And you prepare these amplicons using two different pools of primers. And the amplicons are around 350 base pairs. And after the amplicon libraries are generated per sample, the samples are multiplexed together. And in this instance, I did 24 samples in one sequencing reaction. They're barcoded and then adapter ligated to run on a nanopore sequencer, in my case, the GridION. And on the right here, what I have is a summary of the CT values for all the patients. So out of the 120 patients, I've just taken a small sample of 10 to validate the pipeline that I have. And one thing to note is that the CT value of a viral RNA sample is inversely proportional to the viral load. So a low CT value correlates to a high viral load, which is why I flipped the axis on here so you can, it's more visually intuitive. So you can see that on the low CT values for each patient towards the high CT value of each patient, over time, it does go down as what you expect through a course of infection. And additionally, I have used the low CT values, therefore high viral load, as a proxy of the initial sample given from the patients and the next and then the highest CT value as a later sample, because I am at the moment blinded to treatment arms and also the days of the samples. This is the biophatic pipeline that I use. So once the sample is sequenced on the GridION, the GridION work, the nanopore sequencing works by, as the amplicon goes through the membrane, a signal is given that indicates what base is given, what base is at that position. And so an algorithm is used to call those bases and provide a list of reads. The reads that pass the quality control then go on to get demultiplexed, so that I have them as individual samples again. And then those reads have the chimeras removed. So chimeras can form when primer dimers form during the amplification reaction. And so it's important to remove those. And then the filtered samples will then go through the Arctic network pipeline, which consists of aligning the genome back to the original reference genome for SARS-CoV-2. And the primers then get trimmed. The reason the primers get trimmed is that where the primers bind along the piece of RNA, that they may mask variants and also they can mask errors. So we remove those. Removing the primer regions doesn't matter too much because the primers are tiled across the genome. There's loads of overlapping regions, so we don't miss any information that way. From the primer trimmed alignments, we then generate a consensus of all the reads from each patient, of the consensus of the virus that was present in the patient at the time, using the default settings. And once I get the consensus, I can then run the consensuses through these two programs, Pangolin and Nextclade, which will generate the lineage assignment for the variants of SARS-CoV-2. In parallel, I use the alignment and the primer trimmed alignments to look at interpatient variation analysis using a program called Diversitools, which looks at the nucleotide variation, also the amino acid variation. And in this presentation, I'm going to focus on the amino acid level variations. So what we have here is after I've done the Diversitools analysis looking at the amino acid variation, I've got the two sets of samples, so the patient's first sample and then their sequential samples. And I've plotted the mean non-synonymous amino acid change frequency per protein. So along the bottom, we have all the proteins along the length of the genome, and each row is an individual patient. And what I've highlighted here from the Nextclade lineage assignment is that 8 of my patients were infected with the Delta variant, and 2 of the patients were infected with the Alpha variant. And what we can see here is that we do seem to have a pattern of variation that's separated by lineage, as we would expect. So I can highlight this, but I can highlight this by looking at these 3 proteins. So ORF7A, 7B, and 8 are relatively small proteins, but they do have these dominant amino acid changes that are present in differentially in the two variants. So as you can see in ORF7A, we have these 2 dominant amino acid changes that are present in Delta, but not in Alpha. And you can see that reflected in this data here, where you have this higher mean frequency in the Delta in this protein and less in Alpha. The same goes for ORF7B. And ORF8 is the reverse. So Alpha has 3 dominant amino acid substitutions. And so we have this very bright signal because ORF8 is a very small protein, whereas Delta only has 2 and has a slightly less strong signal there. So this is just to show that using my pipeline, I can identify expected changes to see, and they separate out by variant. And which is useful because when I do the further analysis, if there are any unexpected changes, I should be able to see them as these bright signals of green elsewhere in the genome. And this is a summary of some common amino acid changes I identified, and I've filtered them out by being dominant amino acid changes that are not lineage defining, so that each of the variants have defining amino acid changes. And so what I've included at the top here are some lineage defining amino acid changes that are present in both in Delta only or in Alpha only. And I get the expected frequency. So all 10 patients have the NSP12 amino acid change, only the 8 patients who are assigned as delta have the membrane amino acid change, and the same for alpha. And what this is just to show is that I then look up these amino acid changes on GISAID, which is an international database of SARS-CoV-2 sequences, just to see how common they are. Many of them are uncommon or unreported, and some of them are fairly common across sequences collected worldwide. And what I can use this for is a way to do further experiments in vitro using reverse genetics to see if any of these amino acid changes have a functional purpose for the virus. In conclusion, I've shown that it's possible to identify rich amino acid variant level and genomic data from nasal swabs using publicly available programs. I've also identified predictable regions of high variability across patients that allow me to visualize the differences in the variants that the patients were infected with. I also identified unique amino acid changes not previously reported on GISAID that I can use for further validation in vitro. And I've also established a foundation pipeline that I can use for more detailed analysis in the future when I become unblinded to the treatment arms and can do a larger cohort-wide investigation. And with that, I'd like to thank my supervisors, Julian, Tom, and Miles, and the lab members here in Liverpool that have helped me massively with when I started in the lab, both practically and also with my bioinformatics. A special thanks goes to Katie and William at the GCP labs for coordinating and providing me the samples from the trial. And of course, major thanks go to the Agile partners who created this consortium and allowed for the rapid testing of novel antivirals, and thank, say, the chief investigator and the Liverpool team from the Liverpool site, and my funding. And I welcome any questions. Thank you.
Okay, thank you, Aya. We have one question from the audience, and if any other audience members have any questions, you can put them in the chat now. But this question is, have you found anything interesting in terms of unreported amino acid changes? And are there any hypothetical functional consequences of these changes?
So not anything further than just identifying them in my data set. So I haven't, what I plan to do is in the next stage of my PhD is to do some functional analysis. So I can reverse engineer those amino acid changes and just in the single proteins and see if, for example, if I look at transcriptomics in cells that express that protein, do they interact differently with host proteins? Or I could express them, or I can do sort of like binding assays and things with different proteins like that. But I think that's definitely a next step that I plan to take. This is a very quick snapshot of my data set to see if my pipeline works.
Really interesting work. Thank you, Aya. So we're gonna move ahead to Tony Bell Kareem. Tony Bell Kareem is a student at Tulane University School of Medicine, and she's going to talk about the Lassa-related viruses, cross-reactivity, and its impact on in-field diagnostics.
Good morning. Good morning. Let's see. So yes, good morning. My name is Antoinette Bell, or Tony, and we'll just talk about what I've been doing for the past 3 years with Lassa and its fellow Arena Viridae family members, and how those implications of possible cross-reactivity in our in-field diagnostics have caused questions on whether we actually see Lassa and what our surveillance looks like for our information. So just to kind of a little brief description on what Lassa is, it is a RNA virus. RNA virus that's small, and it has glycoproteins on the outside, and then 2 segments of RNA on the inside, the large and small segments. And we are looking at the small segment, which is the sRNA here to the left. And so on that segment, they have a nuclear protein, which is going to be utilized for a lot of activities since we have a small virus. So it is part of the Rhinoviridae family, but also a old world complex, which just describes where it originated from, being in Africa. So about this virus is that it is pretty deadly, which is not awesome, but as an infectious disease virus virologist, it's the awesome part. But some of the symptoms we see originally when it was founded in found or discovered in 1969 in northern central Nigeria, they saw a lot of hemorrhaging and essentially what looked like Ebola. So not only would they have to kind of confer whether this was a new virus, but also try to figure out if it wasn't Ebola, malaria, and some of the other endemic viruses in West Africa, more importantly, Nigeria. Now we know a little bit more about it, and it has been seen in about 10 different other West African countries, also abroad in the UK and America. Its reservoir, or where it, for a virus where it sits in a person or an animal or even a plant and doesn't harm it as much, should we say, is a field mouse, the mastomy. This mouse can actually transmit the virus because Lassa is zoonotic, either through people eating the animal or through it actually defecating and urinating around food sources. So for this research, which is based on cloning and expressing proteins and looking at antibody interaction between these proteins and how that comes together like an intersectionality with everything for diagnostics. We want to take a look at Lassa in specifics, but also 3 major clades or strains, if you would call it, for Lassa. Clade 2, which is in red, clade 3, which is in the cute green color, and then yellow, clade 4, which is major part where we have a satellite lab in Sierra Leone before the world closed down. This is where we were getting all of our samples, working with patients in one-on-one. Nigeria, we have a few collaborators there, so we're able to get samples, but these 3 clades are of importance. They're one of the top 3 major infections that we see, infectious clades. We also have 4 to 5 others that are new and ongoing. We're always coming with new clades. So these are the ones that we're gonna look at today. And then also, we're going to look at 3 different viruses. So we call these the Lassa-related viruses. Essentially, I think of them as cousins to Lassa. Lassa is pathogenic. However, these 3 are not, so non-pathogenic. We have a virus from the Central African Republic that was found in 1970. We have Mobala, which was found in the later 70s, also from the Central African Republic, and then virus, which was in 1977-ish, found on the southeast part of Africa. The reason we call them cousins or Lassa-related viruses is due to the genetic similarities we've seen through phylogenetic analyses. So we kind of place these together with Lassa, and we study them comparatively, and you'll see that here. So as I was saying, the small segment RNA piece has the nuclear protein. This protein, because the virus is super small, you need to have multiple jobs. So this protein is actually the most abundant protein you'll see during infection, but it also is utilized when the virus is a whole particle as a protector for that RNA. So it's like a pearl bead with the RNA inside. So it helps to support that RNA during travel and everything of the virus particle getting to its host, but also once inside after entry, it will help in replication, transcription, and also suppression of interferons. So we're definitely going to look at this nuclear protein and actually clone this nuclear protein from those 3 Lassa-related viruses. So clone them and then also express them, of course. So the main significance of this research is to take a look at what we have now for diagnostics in the field, in our diagnostic chain, right? So in the field, which I'll explain a little bit later, is a rapid diagnosis test. It kind of looks like a pregnancy test where you place a sample and you have things that'll collect and give you a positive or negative. Once you have a positive, you will bring it to a lab, a proper BSL-2 lab, and do an ELISA to see what you have, any antibodies or anything for your sample to make sure you have a positive. And to even double check, you'll even maybe do a RT-PCR. That's not my ministry, so we're not going to talk about that today. But this is the chain that we are working with. Currently, there are no commercialized diagnostics. We are utilizing research-based things that a former lab member of mine created, the RDT, and we just try to work with that and get as best as specific and as we can, so that we can make sure we are informing our patients for loss of surveillance. So initially, I was just working with IPI virus. Before my PI wanted me to do more. And so I'd already expressed and cloned the nuclear protein for this virus and coded it on an ELISA plate, making my own ELISA. And I took blood from our Sierra Leonean satellite, and we had samples. So I already knew whether these samples were loss of positive, negative, or indecisive or inconclusive from an ELISA test. And I was actually surprised to see the cross-reactivity. There are old 1985-ish papers talking about the cross-reactivity between Lassa and some of the Lassa-related viruses, but not much has been done in this in the current last 5 years. So seeing such cross-reactivity, I wondered what impact that would be on our actual in-the-field diagnostics. This is RDT that we're talking about, the rapid diagnostics test we would use in the field. It's super cute. It's super quick, 15 to 25 minutes. You place that sample on that red spot and it goes up and it'll tell you a positive. So you get a 0. We're not even looking at zeros or ones, but if it hits 2, we will take it and go to the next chain of the diagnostic test. So I took these proteins that I worked, IPI, Mobala, Mopia, these 3 loss-related viruses, and looked at the most specific RDT that we have, which is just for Sierra Leone where we work. And everything was good in the fact that everything was negative. However, when you look at our pan Lassa, which has the antibodies inside of the RDT for clades 2, 3, and 4, the majority clades, you see some more positive tests. Now, if we would see this in the field, we would then take it to ELISA for the next step of your diagnostic chain. And looking at IPI, I kind of figured out already that IPI is most cross-reactive with clade 3, and that was the same I saw for Mobala and Mopia. Then breaking them down even further in their own ELISA. Where's my piece? All right. Them down in their own ELISA work, their own ELISAs where I coded them with their own respective nuclear proteins. We saw that clade 3 was most cross-reactive, following with clade 2, then clade 4. And you'll see just based on your ODs here, which is your signal from ELISA, that Mobala was actually the least cross-reactive, which kind of goes along with the genetic sequencing and phylogenetic analysis that we have seen before. So that was the most generous ideas that we had for the diagnostic test, and we wanted to take it further by breaking up these proteins into even smaller pieces to see if parts of it was more immunogenic than other places. So that's the next step of that journey. Other projects that we have in this lab is we're looking at what is the mechanism between Lassa virus infection and miscarriages that we see with some of our patients. Some patients who have severe Lassa virus, we actually have to, not we, because I'm not that type of physician, but physicians usually have to evacuate the fetus to try to save the mother. So we want to look at what elucidate more mechanisms for that, looking at a certain entry receptor, the alpha dystroglycan, utilizing another arenavirus LCMV. So this is my cute little lab. And our PI, Dr. Robert Gary, who is a world-renowned emergent virologist who worked with Ebola and other large viruses. And here are my ways to get connected. And obviously, like other PhDs, I still know nothing and I'm almost about to graduate. So thank you guys so much. I truly appreciate you for sitting down and listening to my 3 years of stress, worry, and love.
All right, thank you, Tony. It may feel like you know nothing, but you know
Yeah.
a lot. That was a great presentation. And if you have any questions, let me know in the chat. Write them out. Quickly, just a kind of a tangent about the Lassa-related viruses. Do they have the potential to become pathogenic?
So they are super pathogenic to mice, which is also really confusing. It might, because the natural reservoir and all of them are field mice, but I think certain mice like lineages are different than African field grass mouse. So they are super, like if you take a regular black 6, it will kill it. But they could possibly have the potential, being an RNA virus, as we see with COVID, coronavirus, SARS-CoV-2, just a few mutations can cause a big issue. And as you can see, all of these arenaviruses in the old world complex can get in through this alpha dystroglycan receptor, which we have in almost every cell. It's very common. So that makes you even more scared. But yeah, so there is always an option with RNA viruses.
Right. All right, well, thank you. Next up, we have Dr. Jonathan Lynch, who's a postdoc at the University of California, LA. And he's gonna talk about exploration of the gut bacterium that imports serotonin, modifies bile acids, and alters host lipid profiles. So with that being said, take it away, Dr. Lynch.
All right, can y'all see my screen okay? I think it should be full screen now. All right. Yeah, good. Thank you so good. Great. Thank you so much for being here. I'm really excited to talk to you today. My name is John Lynch, and as was just introduced, I'm a postdoc in the lab of Elaine Shao. So I think especially for events like BlackInMicro, I'd like to start off with a little bit more formal introduction. So if someone were to walk up to me on the street, I wouldn't necessarily show this picture for too long because they might be mean to me, but I would like to probably answer the question of who are you with something along the lines of, I'm a Black microbiologist. But as a microbiologist, I have to kind of come clean and be a little more honest, which is numerically and sort of organismally, it's a little more accurate to say that I'm mostly microbes. So we've kind of known this for a long time, but we're appreciating more and more that humans are really sort of walking, talking microbe farms. We're covered inside and out with this really dense, complex community of a great deal of different types of microorganisms, collectively referred to as the microbiota. So numerically, like I said, John Lynch is mostly better drawn versions of these different microorganisms. So one thing that's really important to note is that this isn't just something, the microbiota isn't just something that lives on and in us, but realistically, we're understanding more and more that every day, that the microbiota is really, really fundamental and important for physiological and health and disease at all kinds of different body sites. And that's not just in humans, that's within all animals and all organisms, really. So probably the best studied microbiota, at least body-specific site microbiota in humans and in most mammals, is the gut microbiota, or the community of microorganisms that lives within the gastrointestinal tract, where really the numbers and complexity of microorganisms is kind of astronomical and almost difficult to believe sometimes. But the composition and function of the gut microbiota specifically has been associated with lots of different health and disease states. Importantly, not even just within the GI tract or around sort of the functions of the GI tract, but at all body sites. So that goes from the nervous system, the immune system, metabolism, even things like bone development and maintenance. So this is a little schematic showing some diseases that have been associated with specific activities of the mammalian gut microbiota. For the purposes of this talk, I'm going to focus specifically on the connections between the gut microbiota and obesity, or host sort of fat metabolism and maintenance. Now, one way that people can study a really complex and dynamic system like the gut microbiota is to basically perform community profiling. So sometimes, actually oftentimes what people will do is they'll sequence a specific sort of commonly shared segment of DNA or RNA, specifically in this case, the 16S ribosomal RNA gene. And they use that as sort of a fingerprint to identify taxa that are either enriched or higher, more or less abundant in certain disease or health states, to try to get an idea kind of how communities like the microbiota, specifically the gut microbiota, might shift as people either transition between disease states or sort of have and maintain certain diseases. So a few studies in the past couple of years have looked at 16S community profiling of fecal microbiotas from patients who are either experiencing obesity or are non-obese, and you can find certain taxa that seemingly are enriched in obese patients. So for the purpose of this talk, I'm going to focus on this group here. So this is a little taxonomically contrived, but specifically, this group here found that in obese patients, the fecal microbiotas were enriched for this genus of bacteria called Tricibacter. So it's important for me to mention that the reason these are low contrived is because Tricibacter is really understudied, and there's really only one isolated bacteria that's from any of these taxonomic groups, specifically Tricibacter sanguinis, which I'm naming up here at the top of my slide. Now, interestingly, around the same time, another group found that 16S profiling, so find value for 16S profiling the exact opposite pattern, where in this case, they found that Tricibacter sequence abundance was higher in non-overweight patients and lower in overweight patients. Now, I'm not gonna lie, this is kind of happening at the same time as we were finding something else really interesting about Tricibacter, but seemingly unrelated at first. It at least piqued our interest enough to check in deeper. That was specifically work from a former postdoc in the Shao lab, Thomas Fung, who found that this one isolate, or this one species of Tricibacter from the Tricibacter genus, Tricibacter sanguinis, specifically it's the type strain MAL361. At least looking at the genome, it was predicted to have a serotonin transporter. So importantly, Thomas found it's not just a sequence homology to the mammalian serotonin transporter, but Tricibacter sanguinis MAL361. Again, this type strain, the only really studied isolate of Tricibacter sanguinis, encodes a functional serotonin transporter. Importantly, this seems to be not just involved in importing serotonin, but potentially involved in mediating sensitivity of Tricibacter sanguinis to drugs called SSRI, or selective serotonin reuptake inhibitors. Now, these are often the active compound in antidepressants like Prozac here. And what Thomas found was that basically this compound or compounds that block activity of this transporter in Tricibacter sanguinis inhibit both growth and colonization of Tricibacter sanguinis. Now, importantly, the reason it sort of links back to those previous studies I showed you about the connections between Tricibacter and obesity, Thomas also found that Tricibacter colonization in the gut was really kind of powerful for shifting the lipid profiles of mice. So what he did essentially was he colonized germ-free or notobiotic mice, either with this one type strain of Tricibacter sanguinis, that's indicated up here by the green bars, or no microbiotis, so germ-free mice, essentially mice without any microbial associations up here in the blue, or a complex microbiota indicated here by these red bars. What he found, and this is just a heat map looking at the sort of relative abundance of different lipid species in the serum of these mice. You don't need to necessarily read the individual identities of these different lipids, but I really want to just highlight that colonization, monocolonization with this one isolate of Tricibacter sanguinis leads to a dramatically different lipid profile than either germ-free status or a complex microbiota. So this kind of relates back to the previous work that I showed you, and that Tricibacter, at least some Tricibacters, seem to be really important, or at least capable of modifying the host lipid status. So that leads me to kind of the two major questions from this talk today. So one, it seems like there's a connection between Tricibacter, some level, and host adiposity and sort of lipid profiles. I should also mention this isn't just showing up in mice and humans, but it's showing up in other sort of, especially feedlot animals, where Tricibacter colonizes their guts also. But it doesn't seem like there's a consistent pattern. It's not like, the more turi you have, the more fat these animals acquire, things like this. So can we explain some of the diversity or some of the difference in between those different conditions while still figuring out how exactly turi might be linked? And then secondly, and this is kind of the more mechanistic question, but if we do find an association that we can explain, can we get down to sort of the nitty-gritty and the mechanisms of how exactly turi is influencing or responding in some way to host lipid profiles? So to start at the beginning, what we did essentially was we gathered a bunch of different Tricibacter sanguinis isolates from different host environments. We sequenced the genomes and we did a really basic genomic comparison. So just looking at overall sequence identity. And we found was that within the Tricibacter sequence or Tricibacter sanguinis species designation isn't just one homogeneous group. There's actually a decent amount of sort of subgrouping in there. So essentially we can find 3 distinct subgroups of Tricibacter sequences, again, just looking at that one sort of fingerprinting 16S RNA gene. Now, interestingly, between these three subgroups, two of them are from human isolates. We essentially have two subgroups that are human and really closely related sequences. And then the other group comes from mice. So there's not surprisingly in the sort of evolutionary differences between isolates from different host backgrounds. Now this kind of leads to a really simple and straightforward experiment. What we can do now is we took gnotobiotic mice, so mice that initially have no associated microbes. We monocolonized them with individual strains or individual isolates of Trisibacter from these different subgroups. So you don't need to necessarily remember the names of these, but just, I'll be using these colors throughout the rest of the talk. And I'll keep a little diagram in the corner for you to reference. But essentially we have one isolate from the sort of top human subgroup, two from this middle subgroup, and then one isolate from the mice. So we monocolonize with these individual isolates. And then after 2 weeks, we measure some sort of basic lipid phenotypes of these mice to see how these different isolates might be influencing or not influencing these different phenotypes. So one of the first things we looked at was just how much fat did they gain. This is all in comparison. This is looking at basically the mass of the major fat pads in the mouse's abdomen compared to germ-free mice. So this is all baseline to the germ-free litter mates of these animals. So importantly to note is monocolonization with 3 of our 4 strains leads to an increase in fat pad accumulation, actually to kind of an intermediate extent to if the mice were given a complete microbiota. But colonization with this green H121 isolate doesn't seem to influence the same or doesn't induce the same level of fat acquisition. In some cases, it actually might lead to a slight reduction in fat acquisition. If we do metabolomics on the serum of these mice, so the circulating blood, not just necessarily in the GI tract, but in the entire mouse's body, we also see some really some interesting other lipid-associated phenotypes that are sort of species or strain-specific, and that colonization or monocolonization with individual tertiary strains generally leads to a decrease in cholesterol levels of these animals. But that's the magnitude and the sort of significance of that varies based on the isolate. So basically, this human strain, these mouse strains both lead to significant decreases in cholesterol relative to germ-free animals. But colonization with these sort of middle group human strains don't lead to quite as dramatic a decrease in cholesterol. So essentially what I'm describing, and actually something we've heard about a little bit, is that there's sort of microbial diversity within the Trisibacter sanguinis grouping that might be playing some sort of functional, or might be revealed in functional outcomes relative to host lipids. And that some strains lead to an increase in fat tissue, whereas some don't. And then some strains lead to a decrease in sort of lipid and lipid-associated molecules like cholesterol, whereas others don't. So essentially what we have is that, kind of straightforward question, or straightforward answer to a straightforward question, but essentially different isolates or different strains of Trisibacter sanguinis have different effects on host lipids. Okay, so that's fine. And it's, again, not surprising to see microbial diversity. But we wanted to try to understand exactly how this might be going on. So what specific factors from these different isolates might be explaining these differences. To do that, I need to kind of take a step back and explain one thing that is actually known about Trisibacter sanguinis. So about 2 years ago, Federico Rey's lab found that this type strain of Trisibacter sanguinis, so MOL361, the one that I mentioned earlier, that's the only really studied isolate. This is actually really good at modifying bile acids from the host. So bile acids are produced by lots of different animals. Typically, they have sort of base structure. So on these 2 diagrams, it's this cholic acid base here on the left in black. There's lots of different flavors, but they're produced by the host in the liver, stored in the gallbladder, released into the small intestine, to typically thought to aid in digestion, specifically digestion of like lipids and fats, acting as emulsifiers. One thing that a lot of hosts do is that they conjugate these bile acids to amino acids. So in this case, I'm showing you is a taurine-conjugated cholic acid. Essentially, you can add an addition of a taurine to the cholic acid base structure of this bile acid. And then its sort of counterpart, which is a glycine-conjugated bile acid. So what these typically do is that they might modify a little bit of the functionality of these bile acids, but they're also thought to be really important for sort of changing solubility of bile acids. Now, this is something that happens on the host side. The thing that Turi does, or the thing that MOL361 does, is that it's really, really good at modifying these bile acids and sort of chemically altering them. So specifically, the thing I'm going to talk to you mostly about today is that MOL361 is really good at deconjugating these bile acids and specifically clipping off the taurine or the glycine, reverting them back to sort of their base bile structure. Now, I mentioned, I don't have time to show you a ton of data for this, but one thing that's really important is we didn't know if that was something that was specific only to this one isolate of TERI, or if this is sort of a capacity or capability that was shared across all these different strains. So what we did was essentially we exposed the individual isolates of Theresiobacter sanguinis to a combination of conjugated bile acids and essentially saw what came out the other side, so what modifications they could make. We found, actually this is fairly surprising to me, but we found that basically not all TERIs had the same modification capacities. Specifically, this sort of subgroup 1 of human isolates has a really broad acting capacity to modify and deconjugate bile acids, specifically by deconjugating both taurine and glycine conjugated bile acids. However, this other subgroup of human isolates preferentially deconjugates glycine-specific bile acids, while essentially leaving taurine conjugated bile acids alone. And then these mouse isolates do the exact opposite. So in that case, they can preferentially deconjugate taurine conjugated bile acids while essentially leaving the glycine conjugated bile acids alone. So essentially our general sequence phylogeny is ending up being reflected in a lot of the capacities of these different individual isolates or subgroupings of isolates. Now, I mentioned that this is just kind of, one thing I didn't mention actually is when we sequence these genomes, we found there's a huge amount of just general sequence discrepancy between them. So something like 0.8 megabases difference between different isolates. So we really want to track down and see if we can identify which specific sequences or genes might be associated with individual capacities. So what we did is we looked at our annotated genomes and tried to identify genes specifically involved in bile acid modifications. And for this, I'll show you just some genes that seemingly might be involved in deconjugation of bile acids. We found if we look through these different genomes, I'm color coding by the genome they came from, but each of these represents an individual potential bile deconjugating gene, or bile deconjugase. We found that actually these sequences also seemingly reflected really well, or seemingly mirrored our general phylogeny. So essentially, we see the same subgroupings for most of our bile acid deconjugases. The one exception is this sort of outlier bile acid deconjugase that's shared between these two mouse isolates and has a close homologue and are broadly deconjugating and modifying MOL361 type strain. So we took these individual genes, we cloned them into bacteria that don't really do much biomodification. And then we tested the activity or the specificity of individual bile deconjugases to see if we could identify, what's the overall repertoire these original TORI strains might have. And we found that this actually really well capitulates, or this recapitulates the overall capacity of the cells. So essentially, our broadly modifying or broadly deconjugating group of TERI isolates contain deconjugases that broadly act on both taurine and glycine conjugated bile acids. Our middle group actually seemingly prefers the glycine conjugated bile acids, has genes that preferentially deconjugate glycine conjugated bile acids. And then our mouse group, as well as our broad modifier, has a deconjugase that works on taurine-specific deconjugase. So this is really just almost perfectly reflects what we saw with using our native TERI strains. So this isn't a complete answer, but it's something I'm working on right now. Given that bile acids are really important for things like lipid metabolism and general metabolism, and that TERI is such a powerful sort of modifier of bile acids, our current hypothesis is that the connection between Theresiobacter abundance, Theresiobacter activity, and host lipids might be coming from the activity of some of these biomodifications, specifically things like deconjugation, which we know varies between the different isolates of TERI that we found. So in some kind of future / I have an experiment running right now, experiments, one thing we're doing is kind of a mirror of what we did in those initial experiments, only this time we're taking germ-free mice. Instead of colonizing them with individual strains of Theresiobacter, we're colonizing them with bacteria that don't usually do a ton of biomodifications, but that are contained or expressing TERI-specific biomodifying genes. So essentially, we're kind of just adding an addition to a bacteria to test the activity of individual deconjugations or other biomodifications in the absence of sort of the other mysterious TERI black box stuff. And then we're going to measure those exact same phenotypes that I mentioned earlier, such things like sort of phenotypes such as like weight acquisition, fat accumulation, and serum lipids. And my prediction is that different deconjugases and different biomodifications will have sort of differential effects on some of these phenotypes. So we might have some deconjugases, or for example, they don't really have any noticeable lipid effects, but some might have sort of an increase in things like fat tissue and cholesterol levels. whereas some will have the opposite effect. And again, this sort of gets to how does TERI specifically affect host lipid status. And the sort of long-term goal of this, or the big picture goal of this, comes from that thing I mentioned about why we initially got interested in Tricibacter. If what I'm telling you is true, and I think it is, that Tricibacter is sort of critical and important for modifying fat and weight gain in at least mice and probably humans too, that leads to sort of interesting implications for the effects of drugs like antidepressants, specifically SSRI-containing antidepressants. So in this case, if something like fluoxetine, again, the active compound in Prozac, inhibits Tricibacter colonization and activity, that also might inhibit the activity that Tricibacter would normally impart on things like weight modulation and fat acquisition. So one of our goals is to essentially create workarounds where we can basically complement the activity of Tricibacter in a non-SSRI sensitive bacterium to sort of reduce the side effects, the unwanted side effects of drugs like SSRIs. So some side effects that people experience, I think, like weight gain, weight loss, metabolic shifts, and for drugs that are really important, like antidepressants, having issues like that, like having side effects, can really affect things like compliance and tolerance for these drugs. So we want to kind of work around those. And so with that, I'd like to just summarize by telling you a few things that I've sort of hopefully convinced you of about Tricibacter sanguinis. So one is that it modifies host lipids, or at least host lipid status, in a strain and isolate specific way. So some, both at the sort of grosser level, like just tissue acquisition and maintenance, and also some things like in lipids, in individual lipids. Potentially it does this by doing like modifying host bile acids, again, in a very specific, or say strain specific manner. And that this connection between Tricibacter sanguinis and sort of host physiology is really important to understand if we want to understand how drugs like antidepressants might be sort of affecting host physiology overall. And so with that, I'd like to say a big thank you to my advisor, Elaine Shao, who's been amazing for this project and really helpful for me in a lot of ways. Two amazingly talented undergrads, Kaylee Choi and Erica Gonzalez, who have survived COVID intellectually and really been helpful and critical for this project. Some of our collaborators, my funding sources, and of course, the BlackInMicro organizers for really for organizing an amazing event that I'm excited to be a part of. With that, I'd be happy to take any questions.
Okay, so thank you, Dr. Lynch. You have one, well, you have two questions. You have a question from the audience from Mackenzie Gatlin. So I am going to let them answer the question. Okay, Mackenzie. I think you're muted. Okay, well, you have a second question, so we can jump to that one. And that is, what would you expect gut microbiota related to obesity, or would you expect gut microbiota related to obesity to differ depending on where one lives?
Oh, for sure. This is actually one of the things that's really difficult about studying the microbiota, is that it's really complex in terms of community status. So for one, even if you do 16S, you can describe hundreds of thousands of species within the gut microbiota of an individual. That's sort of defined by things like their diet, their location, their health status, also potentially described on like how exactly you're studying it. But it definitely seems like the geography and sort of geography associated things like I guess like drug treatment, or I guess in general like medical treatments, diets, interactions with other people, those can all influence the overall composition of the microbiota, which in turn will influence the activity of the microbiota. So it's really hard, and this is kind of like the golden bullet everyone's looking for. People really want to find like, here is the one thing that causes obesity, here's the one thing that's really important for this disease state, but it's much more likely and seemingly almost inevitable that it's not like a specific microbe that's doing this, and instead is probably an activity or a group of related activities that are important. So I think that depending on where you live, your microbiota might be different, and the sort of outcomes associated with your microbiota will likely also differ.
Okay, and then we have one more question in the chat from Tony, and she wanted to know, do you have other bacteria in mind with Tori genes for weight modulation? And are these bacteria native to the microbiome of the stomach?
Yeah, so to be perfectly honest, right now there's so little known about Terry. I have more than I want to work with there. So for perspective, I've been able to find 3 growth curves since Terry was first isolated in 2002. They've all been with one strain of Terry. So we know almost nothing about any sort of classic microbiology of Bacter besides what it looks like some of the time and its genome sequence. I don't want to imply that I think that Bacter sanguinis is the only modifying or only weight or fat or metabolism modifying microbe. It definitely isn't. I think it'd be really interesting to find patterns that are present in Terry that are also shared with other microbes. Because like I mentioned a second ago, it's probably easier to target something like a functionality or at least a shared functionality amongst a group of microbes that influence something like host metabolism than it is to specifically say, oh, we're going to give you lots of Terry or not give you lots of Terry. So I think as we learn more about Bacter activity and sort of physiology, it'd be really awesome to try to find similarly acting bacteria and other microbes even that are acting either similarly acting microbes that are doing kind of the same thing, but maybe in a slightly different flavor to try to find generalizable approaches to dealing with those issues.
Okay. All right. So Dr. Lynch, you have one more question in the chat. I'm going to ask that you answer her directly in the chat because we're going to move on to the next speaker. So thank you for your talk.
Thank you.
So the next speaker is Jadine Sewell, and she is a student at the University of Pennsylvania, and she's going to talk about CVU 0122, which is a novel Coxiella effector protein. A novel Coxiella effector protein modulates the fusion of the Coxiella containing vacuole. So take it away, Jadine. Are you muted?
Hi, everyone. I'm trying to get my video on. Oh, there we go. Hello, everyone. Thank you for having me. Let's get started. So today, I want to talk to you about this project I've been working on in my master's. And it's investigating CBU 0122, a novel Coxiella effector protein that modulates the fusion of the Coxiella containing vacuole. So Coxiella badeni is an obligate intracellular bacterial pathogen. It primarily infects areolar macrophages, and we humans typically get infected through contaminated dust particles from placental remnants from farm and wild animals. Coxiella primarily infects areolar macrophages, and once it infects the cells, it utilizes the host membrane trafficking pathway. Once it infects the cells, it utilizes the host membrane trafficking pathway, fuses with early endosomes. Upon fusing with early endosome, it further matures into a late vacuole, and this is the Coxiella containing vacuole, which I'll further note as CCV in the presentation. Once it fuses with late endosome, it becomes very acidic, and upon acidification of that vacuole, it activates its .ICM type 4 secretion system to secrete effector proteins within the host cytosol. Upon maturation, it further fuses with lysosome and autophagosomes and form this big vacuolar replicative niche. And while it fuses with host compartment, it accumulates several different host proteins from these membranes, such as RAB7, LAMP1, and LC3. A subset of these effectors we call the coxalovacular effectors, termed CVPs, localized onto the coxalovacuole membrane during infection. CVPA, which is an effector that fuses and that localizes onto recycling endosome and interacts with clathrin, is found on the membrane. CVPB interacts with PI3 enzyme and localizes these lipids onto the membrane, and it also interacts with causes homotic fusion with the CCVs within the cell. CBPF, which is a new effector that was newly identified, interacts with autophagosome, fusing these autophagosome membrane to the coxiella containing vacuole. I further want to talk about this newly protein that we identified, CBU122. So during infection, CBU122 localizes onto the coxiella vacuole membrane. As you can see here, we have the host membrane nucleus in blue, GFP expressing CBU122, and coxiella in red. And we can see during an infected cell, CBU122 is localizing around the coxiella vacuole membrane. However, in uninfected cells, CBU122 localizes to the perinuclear regions of the cell. Further looking at CBU122 protein, when CBU122 is ectopically expressed in cells, we see an increase in the number of CCVs within the cell and a decrease in the sizes of the vacuole. We also see a change in different proteins that are during when wild-type coxiella infection, when we express GFP-RAB, which is a recycling/Golgi associated protein, we see that the coxiella vacuoles are regular VIC vacuoles within the cell, and we see a decent amount of RAB9 protein accumulated around the vacuole. However, without this CBU122 protein present, we see an increase in RAB9 association within the coxiella and around the coxiella containing vacuole. So what I wanted to do was further to figure out which host compartment CBU122 is localizing to when it's secreted into the host cytosol. The methods of my project and what I wanted to do overall was to plate cells and transfect them with GFP expressing CBU122, and then fix and stain these cells for host protein markers for the cis-Golgi network and the recycling endosome network. I use GM130 for that that marks the cis-Golgi, and CD71 that marks recycling endosome within the cell. For my first experiment, I wanted to see whether CBU122 was preferentially localized into the cis-Golgi or the recycling endosome. Upon doing this experiment, I could see that I wasn't really sure, or I could tell whether CBU122 was localizing more with the recycling endosome or the cis-Golgi, because we see an association indicated by the co-localization of the red, which is indicated of the GM130 or CD71, and the GFP expressing CBU122. So I wanted to take this a step further and try to separate these two compartments because they're in the perinuclear, both these compartments are within the perinuclear region of the cell. So I wanted to treat these cells with a chemical called Berfeldin A, which disrupts the Golgi network and from a nice stack of cis Golgi to a fragmented Golgi, and disrupt the recycling endosome from puncta into tubulated forms. So I hypothesized that if we dysregulate these compartments, we'll change the CBU122 localization to the perinuclear region will be affected in uninfected cells. So I first wanted to see whether recycling endosomes would be affected with Berfeldin treatment once we disrupt these compartments with Berfeldin A. So I could see before Berfeldin treatment, we see an association with CBU0122 with the recycling endosomes. And as we disrupt these compartments, within 30 minutes of Berfeldin A, we see a tubulated recycling endosome compartment, but we still see CBU122 associated with these compartments. However, this is a little bit different when it comes to the cis Golgi. Here, after repeating these experiments, we see that CBU122 is not associated with the cis Golgi, and upon further treatment of Berfeldin A, which disfragment this Golgi, we can see that there's a clear separation between the CBU122 protein and cis Golgi. I further wanted to quantify this, and I did a single cell quantification, looking at the co-localization coefficient between the recycling endosomes and the cis Golgi marker with CBU122. And as we could see right here, even though after Berfeldin treatment, we don't see a difference between the co-localization of CBU122 with the recycling endosome or the cis Golgi. However, we see that there's a clear co-localization with the recycling endosome compared to the cis Golgi. So in conclusion, we could see that CBU122 localizes preferentially localized to the CCV during infection, but in uninfected cells, we see that CBU122 localizes to the perinuclear region in the cell. Ectopic expression of CBU122 alters the size and increases the amount of CCV within infected cells. CBU122 localizes preferentially to CD71 positive recycling endosome compartment within the perinuclear region. However, disruption of recycling endosome vesicles, we see that CBU122 localizes to TFR marked transferrin receptor marked tubules after BFA treatment. So some future directions from this project would be immunoprecipitation followed by mass spectrometry in vitro assays to identify binding partners of CBU122, or investigate CBU120 effects on transferrin recycle when expressed in the cell. We could also further evaluate whether CD71 localizes onto the CCV membrane, and investigate other host protein markers such as syntaxins, which are localized onto the trans-Golgi network, which when we treat cells with Berfeldin A, recycling endosome tubulates with the trans-Golgi network. So the syntaxins could be a potential place for localizing, CBU122 may be localizing too. And with that, I just wanted to leave this picture to show that Coxiella secrete over 100 different effectors that goes and affect different host functions such as apoptosis. It affects it's localized to the mitochondria, localizes to the nucleus, and it disrupts and localizes with and utilize this host membrane trafficking pathway to fuse with different host compartment and create this big lysosomal vacuole to replicate and survive within the cell. And with that, I would like to thank my former PI, Dr. Shauna Reed, for guiding me through this project, Quinnipiac University, and I will take any questions.
All right, you have one question in the chat. So that question is, what type of protein is CBU122? And what types of domain does it encode? And are there any predicted functions in its annotation?
Yes, so CBU122 is it's a really small protein with predicted trans-domain regions. We don't know a lot about I haven't looked into the structure. We don't know a lot about the structure, but we do know that it predicts to have trans-domain regions, indicating that it could localizing onto the membrane, within the membrane.
All right, well, thank you for your talk. It was insightful. We're gonna move on to the next speaker. who is Dr. Ed Cunningham Oakes, and he is a postdoc at the University of Liverpool in UK. And his talk is titled Better Later Than Never, Identifying Burkholderia Enigmatica as an important industrial contaminant using genomics. So with that, Dr. Cunningham Oakes, you can begin.
Perfect. I'm just sorting some security settings because I need to double check I can actually share this. Bear with me one moment. Okay, it appears we have some technical difficulties sharing the presentation. Would it be possible to just move on a talk and perhaps I can work this out with the organizers just to get this shared?
Yeah, we can do that. 1 second.
Yeah.
Dr. Afiki, are you ready to go? Hello? Dr. Washington, are you ready to go?
Yes, I am.
Okay, we'll do you next.
All right, with that, I will introduce Dr. Washington. So Dr. Erica Washington is a postdoc at Duke University, and she's going to talk about the interactions among trihalose biosynthesis proteins, implications for antifungal drug development.
Great. And let's see, my video won't start. There we go. Okay, hi. Screen. Okay. Great. Are you able to see my screen?
Yes.
Okay, great. Thank you so much for this opportunity to present at Black in Microbiology in 2021. I'm excited to be able to share my research with you that I've been doing in Richard Brennan's lab in the Duke Biochemistry Department on complex formation among trehalose biosynthesis proteins and our interest in developing an antifungal drug target. So invasive fungal infections are a major threat to human health. The invasive fungal infections result in more than 1.5 million deaths worldwide each year. And these infections commonly occur in immunocompromised hosts. And these include patients that are on immunosuppressive therapies or patients with HIV/AIDS. More than 90% of all of the reported fungal-related deaths result from species that belong to 4 genera. And these include Cryptococcus, Candida, and Aspergillus. Now, the 3 major antifungal drugs are the polyenes and the azoles and the echinocandins. And unfortunately, all of these drug classes have limitations. Some of them are highly toxic, and also none of them have broad spectrum antifungal activity. In addition, with the emergence of new fungal pathogens and antifungal drug resistance, it's really important that we develop new antifungal drugs. So how do we identify targets? One way is to target the ability of the pathogen to grow in multiple environments. For example, for a fungal pathogen to survive in the human host, it has to do a temperature jump from the ambient temperature of the environment to 37 degrees in the human host. The trehalose biosynthesis pathway in fungal pathogens is a critical factor that is required to make this jump. So what is trehalose? Trehalose is a non-reducing disaccharide that's made up of 2 glucose molecules. And it was the genes to generate trehalose were first identified in a set of genes that are expressed during the Cryptococcus neoformans cerebrospinal fluid infections. Now, trehalose can play multiple roles in the fungal cell. It can act as a reserve carbon source, and that it can be broken up into 2 glucose molecules and then funneled into the glycolysis pathway. It can act as a stress protectant by protecting proteins and membranes during time of stress, and it also plays a role in virulence. What's important for our ability to use trehalose as an antifungal drug target is that it is synthesized in bacteria, fungi, and plants, but not in mammals. This gives us the opportunity to develop an antifungal drug that is safe and not toxic, which is important for the immunocompromised patients that are often infected with antifungal drug with invasive fungal infections. So the canonical scheme for trehalose biosynthesis in pathogenic fungi is simple. It starts off with UDP glucose and glucose 6-phosphate, which are then converted to trehalose 6-phosphate or T6P by an enzyme called TPS1, and that releases UDP. In the second step, trehalose 6-phosphate is converted to trehalose with the removal of the phosphate group by TPS2, which is a phosphatase. Also important for our ability to use trehalose as the antifungal drug target is that disruption of TPS1 and TPS2 results in strains that are fungicidal. And disruption of these genes result in strains that will not grow in the host, which makes it a really great antifungal drug target. So we know a lot about the domain organization and the structures of these proteins. TPS1 is a single domain protein, which is a synthase. And so here I'm showing you TPS1 from Candida albicans bound to UDP glucose. The N-terminal lobe and has a C-terminal lobe that's connected by a C-terminal alpha helix. TPS2 has an N-terminal pseudosynthase domain that is similar to the TPS1, except for it lacks the residues required for ligand binding. And so it's inactive. The C terminus of TPS2 is a phosphatase. And so here I'm showing you the Cryptococcus neoformans TPS2 phosphatase domain in a catalytically inactive form, which is closed and bound to T6P. Now, some fungal pathogens have a third protein, which is called TPS3, which in its N terminus has a pseudosynthase domain, and in its C terminus has a pseudophosphatase domain. So there is no known enzymatic activity for TPS3. And so we hypothesize that TPS3 is involved in the formation of a complex among trehalose biosynthesis proteins. So is there an actual complex that's being formed? In other organisms, there are complexes and interactions formed among trehalose biosynthesis proteins, such as Saccharomyces cerevisiae. In addition, as I mentioned, TPS3 has no known enzymatic function, so perhaps it's acting as a scaffold to help TPS1 and TPS2 bind to each other. Additionally, E. coli OTSA, which is the E. coli TPS1, which is not able to interact with TPS2, can also not complement a TPS1 knockout strain, suggesting that the formation of a complex is important for the function of trehalose biosynthesis proteins. And so we hypothesize that perhaps if there's just TPS1 and TPS2, such as in Cryptococcus, that TPS1 and TPS2 interact through the N-terminal domains. And in Candida albicans, where there's TPS3, the TPS3 also interacts with TPS1. So to determine if there are interactions among the TPS proteins, we set up a bacterial 2 hybrid system. And in this system, if your protein of interest dimerizes, then that leads to dimerization of the LEX-A DNA binding domain, which then leads to repression of the LEX-C gene, which we can then test for using a beta-galactosidase assay. So using this this, this assay, the bacterial 2 hybrid assay, we were able to show that TPS1 homodimerizes, but TPS2 and TPS3 do not. Additionally, TPS1 can interact with TPS2 and TPS3, but TPS2 and TPS3 do not interact with each other. And finally, we were able to show that the intramural domain of TPS2 is sufficient to interact with TPS1, leading to our model that, at least for Candida albicans, TPS1 proteins self-associate and can interact through the intramural domains of TPS2 and perhaps even TPS3. So our next goal is to solve the structure of this complex. And these proteins are large enough that it would be difficult to use crystallography methods to solve the complex. So we decided to use cryo-electron microscopy. So can we use cryo-electron microscopy to solve the trehalose biosynthesis complex? We decided to start off using the Cryptococcus TPS1 in our cryo-EM studies. And we did this because the Cryptococcus TPS1 has 3 unstructured domains in the N-terminus and the C-terminus, and then also in the middle, there's a large internal extension loop that is unstructured. These 3 domains have made it very difficult to solve the crystal structure for Cryptococcus TPS1 compared to the structures that we have for the Aspergillus and the Candida albicans TPS1. We're also really interested in determining what the role of this internal extension loop is. And with this protein being 74 kilodaltons, if it does form a homotetramer as expected, then we should be able to detect density for this complex using cryo-electron microscopy. And so we started off solving the structure of the APO Cryptococcus TPS1 cryo-EM structure. So on the left, I'm showing you our map of the density of the APO Cryptococcus TPS1 homotetramer at 3.3 angstroms. And then on the right, I'm showing you the ribbon diagram of the model that I built into the map, showing you that there's 4 protomers and that it in fact does form a homotetramer. So next we wanted to see if we could get a higher resolution structure with a substrate bound TPS1 cryo-EM structure. And so the substrates that we chose were the glucose 6-phosphate, which is an actual native substrate for the Cryptococcus TPS1, and the product UDP. So we incubated our protein with those ligands. And again, we're able to solve a cryo-EM structure. So on the left, again, I'm showing you the density for the ligand bound TPS1, which we were able to get to 3 angstroms. And the ribbon diagram on the right, where you can again see that it's forming a homotetramer with 4 different protomers. And this time, the structure is more compact, and you can see inside of each substrate binding pocket, there's the UDP, and the glucose 6-phosphate. Importantly, the resolution inside of each of the binding pockets reached 2.7 angstroms, which is good enough for us to be able to detect clear interactions of the substrates with our protein. So one of the first things that we noticed when we looked at the 2 different structures of Cryptococcus TPS1 was that when you binding of the ligands induces a complex conformational change in which the ligand bound form closes in on itself. And we can see the majority of the movement occurs with alpha helix 2, in which there's a movement of approximately 7 angstroms inward towards the substrate binding pocket. Additionally, as I mentioned, we were able to see good density for the UDP and the glucose 6-phosphate, which allowed us to look at specific interactions between side chains of the residues and some main chain interactions with the residues and our ligands UDP and glucose 6-phosphate. So we took these specific interactions and these residues and made point mutations to convert them to alanine and then measured the activity of TPS1. So here on the right, I'm showing you the average relative activity of the point mutations compared to wild type TPS1. And what we can see is that each one of these individual residues is critical for the activity of wild type TPS1. So for binding UDP and G6P. Additionally, which is important for us to develop a broad spectrum antifungal drug, is that these residues are conserved in the same binding pocket. So again, I'm showing you the Cryptococcus neoformans in green, and then the Candida albicans residues in gray, and where they're align and bind the UDP and the G6B. Furthermore, in the bottom, I'm showing you an alignment where these residues are conserved all the way to the E. coli OTSA. And so targeting these residues with a drug would perhaps give us an ability to develop an antifungal drug that has broad spectrum activity. And we also wanted to investigate the internal extension loop. And so I'm showing you here that the residues within the loop are quite conserved in the fungal pathogens that have it. And these fungal pathogens include Cryptococcus species, as well as other closely related Basidiomycota. But when you look at the ascomycetes and the mucors, there's no conservation of the internal extension loop, meaning that it's specific to that family. So we wanted to determine the function and whether or not this loop is required for function, since we couldn't see it in our cryo-EM structures. So in Candida albicans, this loop is a small loop. So we made a chimera in which we took the 7 residues from the Candida albicans TPS1 loop and added them to that 91 residue loop that we were missing in the Cryptococcus TPS1. And again, measured the activity of the protein, wild-type protein, compared to the chimera. And what we were able to see is that the internal extension loop is not required for catalytic function in vitro, which leads us to the hypothesis that this loop is important for activity, perhaps in vivo, in non-catalytic function, perhaps protein-protein interactions or post-translational modifications, or even subcellular localization targeting. And so that's something we want to explore in future directions. So we also wanted to look at the specificity of the Cryptococcus TPS1 substrate binding site. And so in order to do this, we used as the substrate in an assay, we used UDP glucose as well as UDP galactose. And the main difference between the UDP glucose and the UDP galactose is just the position of the hydroxyl group on the 4th carbon on the sugar ring. And so when we used UDP glucose, of course, we have wild type levels of activity. And then when we substitute in UDP galactose, again, you see that there's no activity, suggesting that the substrate binding pocket is very specific. And our negative control in this experiment was just UDP without any sugar. So next we wanted to see, does UDP galactose bind at all? And so we did a competition assay in which we measured the activity of Cryptococcus TPS1 with UDP glucose at 1 millimolar, and then added increasing amounts of UDP galactose until we had 10-fold excess of the UDP galactose. And what we were able to see is that the activity of Cryptococcus TPS1 was reduced when you added excess UDP galactose, indicating to us that UDP galactose is binding, but that it's just a weak binder and probably a weak competitive inhibitor. So in summary, we set up a bacterial-2 hybrid system, and we were able to use this to identify interactions among TPS proteins. And we found that the internal domain of TPS2 is sufficient to interact with TPS1. And this was in support of our hypothesis that the proteins form a complex. So next we started our cryo-EM studies, and we solved the structure of APO, Cryptococcus TPS1, as well as ligand-bound TPS1 with UDP and G6P. We were able to confirm that TPS1 forms a homotetramer, which is similar to the model that we developed in our bacterial-2 hybrid assays. And we were able to get high enough resolution in our cryo-EM structures that we could characterize the substrate binding pocket all the way down to specific interactions of residues that are required for activity, as well as looking at movement of the pocket and the specificity of the pocket. We identified a conserved unstructured region in the protein that's not required for catalytic function in vitro, but that leads us to future directions in which we can determine if there's a function for this region in vivo. So in the future, we aim to solve the structures of TPS1 with inhibitory compounds and using cryo-EM again to see where some potential lead inhibitors could bind. We want to solve the structure of the heterotetramer complex of TPS proteins, so with TPS1 and TPS2 and perhaps even TPS3. And to get back to our overall goal of developing an antifungal drug, our goal is to identify compounds that disrupt the complex formation and use this as a way to inhibit trehalose biosynthesis and fungal growth in the host. So I'd like to acknowledge my PI and my advisor in my lab, which is Richard Brennan, and our other lab mates, specifically Hannah McMillan, who was a grad student who worked with me on a separate part of this project, and Yi Mao, who was a grad student who solved the crystal structures of the Candida albicans TPS1. Maria Schumacher is the lab lead of our neighboring lab. And our collaborators on this project are Richard Lee's lab and John Perfect's lab. And I also like to acknowledge our collaborators in the cryo-EM facility, specifically Ye Zhao, who has worked with me on the cryo-EM structures and the funding. So thank you very much. Thank you again for this opportunity to fellowship and be with other Black microbiologists and share our exciting research for the culture. And I'm happy to take any questions.
Thank you, Dr. Washington. You do have a question in the chat from Rebecca Rashid Ackerman.
Okay.
She wants you to know that this is a really lovely study, and she wonders if you could use your bacterial-2 hybrid assay to test for natural products or pharmaceuticals that inhibit the interaction.
So I don't think that the bacterial-2 hybrid will work for looking for compounds interacting with the proteins, but we do have a pipeline of how we are trying to look for inhibitors, including natural products and some lead-like compounds, as well as a bioactive library. And in order to do that, we are using a thermal shift assay that's just looking for binding of the compounds and a change in the stability of the protein when the compound binds. And then from there, we're looking for specific inhibitors that can affect the activity using the TPS1 activity assay that I showed you today. So I don't think that the bacterial-2 hybrid will work for looking for compound interactions, but the thermal shift will definitely help with that.
Okay, and you have one more question from Dr. Jonathan Lynch, and he says he might have missed this, but do you have a hypothesis for what TPS3 is important for? Is it primarily a scaffold for TPS1 and 2, or is it something else about trehalose synthesis in species in certain species with TPS3 sequences?
Yes, thank you. Great question. So our hypothesis is that TPS1 and TPS2 and TPS3 in the species that have TPS3 are involved in forming a complex, and that TPS3's role is mainly as a scaffold. However, TPS3 might have different roles in different fungal pathogens. For example, we know that in Aspergillus fumigatus, the TPS3 is involved with binding and forming a protein interaction with the chitin synthase, and in that way it is targeted towards the cell wall and is involved in chitin synthesis at the cell wall. We have not seen that interaction in any other pathogen except for Aspergillus. So TPS3 might act as a protein-protein interaction scaffolding protein, but it also might play different roles in the fungal cell, such as the chitin synthesis or protein-protein localization, and that's going to have to be teased out for each pathogen. It might be a little bit different. We do know in Saccharomyces cerevisiae, when you knock out TPS3, well, they have two TPS3s, but when you knock out both of them, you have a drastic reduction in trehalose biosynthesis. suggesting to us that it is important, whether it's acting as a scaffold or not.
All right, thank you, Dr. Washington, for a lovely presentation.
Yeah.
So we're going to move on to Dr. Ed Cunningham Oakes, and he is a postdoc at the University of Liverpool, and his talk is Better Later Than Never, Identifying Burkholderia enigmatica as an important industrial contaminant using genomics. So Dr. Cunningham, let me know if you could share your slide.
Okay, can everyone hear me okay?
Yes, and everything looks good. I can see your screen.
Fantastic. This talk is going to be extra ironic because I'm going to be talking about bioinformatics with apparently not being able to use a PC. So here we go anyway. So yes, thank you for introducing me. Also, thank you for Dr. Washington for stepping up whilst I was working out basically how to work a laptop. So yeah, as we mentioned earlier, my talk is called Better Later Than Never, Identifying Burkholderia enigmatica as an important industrial contaminant using genomics. I think I've spoken to most of the organisers by this stage, but I'd like to extend an apology to whoever had to autocorrect that and check that the spelling was correct. So just as an overview of what I'm going to be talking about, I wanted to get you a bit of information as to what my background is. I'm then going to condense basically 20 years of history, going on 30, of gene-based bacterial taxonomy, where we've come from and where we're going. I then want to introduce the Burkholderia cepacia complex as an important group of respiratory pathogens before I focus on the discovery of Burkholderia enigmatica. I'm then going to start using genomics to distinguish species within Burkholderia taxon K. Now, at the bottom of the slide, I've got a publication that's associated with this work. So if you want to look at it in more detail, it's published in Microbial Genomics as of earlier this year. So just a brief overview as to who I am, because I think a bit of context is nice sometimes. I did a pharmacology with extramural degree, which basically means I did a placement year at St. George's Hospital, the Amenhu and Anthony Coates. So some of the work I was looking at there was looking at the susceptibility of Staphylococcus aureus in response to antimicrobials, and specifically using compounds to reactivate antimicrobials, so to speak, that these organisms were resistant to. This led directly into my PhD at Cardiff University, and this is really where the project stemmed from. So the overall project was called the Genomic Basis of Preservative Resistance, and what this was looking at was the underpinning genomics, the whole genome sequencing of bacteria that have grown in industrial products when they're not meant to. So the industrial products that I focused on were home and personal care products with the industrial partner Unilever. And really what we were looking at here are bacteria that tend to grow in products that have fats, oils, and proteins in. So bacteria love those, as anyone who works in the lab will attest. But also these products have preservatives in to prevent the bacteria from growing. Preservatives are quite multimodal in their action, unlike traditional antibiotics. And this means that we actually know very little about what genomically is underpinning the resistance to these preservatives, because they have so many mechanisms of action. So we were hoping that genome sequencing would give some sort of insight into this. Alongside this comes accurate taxonomy, which can be important, particularly in the Burkholderia cepacia complex, as I will describe in a moment. And currently I work within the HPRU at the University of Liverpool, mainly working on metagenomics of diarrhea samples, so a far cry from what I'll be talking about in this presentation. So what is in the name? I want to describe where we've been and where we're going to. So let's start with the 16S ribosomal RNA gene, which I think some of the previous speakers have already spoken about. So this is a particularly useful gene in that it's conserved, and it was particularly seminal in distinguishing bacterial archaea and prokaryotes. And the reason it's so useful is that it's conserved, meaning that it's basically a quick genetic test that you can use to distinguish distantly related bacteria in terms of taxonomy. Although I've alluded to the problem there in that with conservation comes similarity. And the problem with this is that if you have two closely related groups of bacteria, so one good example of this is the family Enterobacteriaceae, where the 16S ribosomal RNA gene can be particularly poor at distinguishing certain species of bacteria, you quickly run into problems if you want to get beyond the genus level. And this is where multi-locus sequence typing was proposed. And essentially what you're looking at in multi-locus sequence typing are a group of 7 genes that are usually allocated by an expert in the field. And usually you're looking within a species or closely related group of species. Now, the way multi-locus sequence typing works is you can either amplify the 7 genes of interest that have been designated, or you can sequence the entire genome and extract these genes. You then compare these genes to a database and look at individual polymorphisms, so base changes within these genes. And by doing this, you can assign each gene an arbitrary and unique number. A combination of 7 numbers gives the overall isolate a sequence type, and you can use this to distinguish bacteria. So how does this relate to the Burkholderia-Sapiecia complex? And really here, what I want to highlight is how the MLST in particular has been used to identify the spread of Burkholderia-Sapiecia complex with regards to cystic fibrosis, basically between Canada and Europe. So firstly, this is a group of closely related species, about 20 or so at the moment, gram-negative bacteria, and the known contaminants of both clinical and industrial settings. They're particularly problematic in cystic fibrosis, particularly when you consider Burkholderia-Sapiecia and multivorans, which are the most virulent in end-stage live in cystic fibrosis patients. And what I've shown on the right-hand side of the slide are a bunch of sequence types that were taken from cystic fibrosis patients, both in Canada and Europe. And what I want to draw attention to in the top left of the continent of Canada and also the United Kingdom is the presence of ST28. This sequence type is also known as ET12 and is one of the most virulent Burkholderia-Sapiecia multi-locus sequence types. Now, the reason this is particularly relevant is that the study that I've highlighted showed that if you pair multi-locus sequence typing with good epidemiological data, you can start to know where to look to try and explain the spread of some of these isolates. As you can see, there's quite a wide geographic distance between them. However, investigation of the patients in particular, a bit further, showed that there was a cystic fibrosis summer camp where individuals who were struggling with disease were allowed to essentially socialize with people who were struggling with the same condition. And this is where the spread began. So MLST is quite developed for the Burkholderia-Sapiecia complex. Now, amongst all of this, a taxon, so a phylogenetic group within the Burkholderia-Sapiecia complex was defined known as taxon K. And this had 2 novel species known as Burkholderia contaminans and Burkholderia lata. Now, the reason this was particularly relevant to the lab I was working in is that in a previous study done by the lab, it was shown that from historical contamination incidents in our panel that was provided by Unilever, the most frequently isolated species within the Burkholderia-Sapiecia complex was Burkholderia lata. However, a 2020 taxonomic study, which used a combination of MALDI-TOF techniques, genome sequencing, and also phenotypic assays, showed that you can actually split this further if you use more genomic information. And this resulted in the novel species Burkholderia enigmatica. And really, this revelation left me looking like poor Obi-Wan at the end of the slide, in that I'd been working with a group of bacteria over the course of my PhD. We weren't sure if they were the correct species anymore. And moreover, we didn't know how to define an appropriate workflow for the identification of this new species group. Now, the reason this is particularly relevant is twofold. The first, which I'm not going to delve into in detail, but I provided a reference at the end of the slide in a review that we did for Femmes Microbial Letters, is that 49% of all organisms recovered from contaminated products that were reported to agencies remain unidentified. So there's a clear identification issue, at least in the non-food industry. Moreover, the accurate identification, at least at the species level, is particularly relevant when you consider that Burkholderia-Sapiecia and multivorans clearly have a species niche in the cystic fibrosis lung, for example, in terms of their virulence. So there can be a clear association between the virulence of species within this complex, despite their close relatedness, and the actual species identity of them. So this slide gives an overview of what we did for this study, but essentially, just to put it into a nutshell, we got 23 historical contaminant isolates that had been identified by MLST as Burkholderia lata, Burkholderia contaminans, and 6 which were just called a novel species within taxon K. These were all industrial isolates, we subjected it to paired end 150 base pair sequencing. And then what we did was compared to all of the available taxon K reference genomes in the database, with the aim of seeing if the greater resolution provided by genome sequencing would provide us any sort of indication as to what the actual prevalence of different species groups was from our industrial products. Because prior to this study, it wasn't entirely clear in that Burkholderia lata can be isolated from the environment, from the clinic, and from industry. So essentially, it's quite ubiquitous. We had MLST types as previous sequencing controls, as a lot of these isolates had profiles generated by PCR, and also the MLST types defined in DePorter et al., which is the taxonomic publication I referred to previously, actually defined which sequence type should be typed as Burkholderia enigmatica now. So what we've done here is done both an MLST and core genome phylogeny. And there's quite a lot going on the slide, but actually the picture is quite clear when you ask the key question, why were the industrial isolates typed as Burkholderia lata, and also what is their species designation now? And what you can see by the multi-locus sequence typing phylogeny in panel A, all of the blue isolates are our industrial isolates, with the exception of Burkholderia lata A05, which is a reference isolate in the database. And you can see that they group quite closely to the Burkholderia lata type strain. However, when you transition to a core genome phylogeny, you'll notice that the Burkholderia lata type strain is actually an outgroup in this phylogeny, and in any other context other than actually showing how different these industrial isolates are to that isolate, it could be considered an outgroup that doesn't actually add a great deal of detail to this phylogeny. But the key thing that's coming out of this is that the Burkholderia enigmatica type strain groups quite closely with all of the isolates that were typed as group K, both in the MLST and core genome phylogenies, although the type strain of Burkholderia enigmata wasn't available when these industrial isolates were initially typed. And the Burkholderia lata industrial strains form an enigmatica-like group. So the key difference between these two phylogenies we're looking at is one that's based on 7 genes and one is based on all of the core genes within the data set, i.e. everything that is in common between all of the genomes that you were comparing in the data set. This is also the case when you use a second technique known as average nucleotide identity. So average nucleotide identity works by essentially clustering genomes in terms of their nucleotide identity. Again, just repeating the name, but what that basically means is that if 2 genomes have 95% or greater identity in comparison to each other, they can be considered the same species. Now, the reason this is particularly useful is you can get useful insights, like you can compare your core genome phylogeny and validate it. You can compare directly to a type strain, and there are several tools that will do this quite quickly now. So this was actually quite quick to generate. And also you get a nice visual representation of the species groups coming out. So anything in red here is over 95% identity. Anything in blue is below 95% identity. And what's interesting is that the Burkholderia lata type strain that is used to designate other species groups is quite unique in the database. There aren't any other isolates like it apart from LMG 6992. And moreover, there are a lot of isolates in the database that are termed lata, contaminans, basically the works, that are actually mistyped and belong to a, as of yet, undesignated Burkholderia species group. But most importantly, our industrial isolates, once again, are designated as this enigmatica and enigmatica type groups. So in conclusion, this is essentially where we were with multi-locus sequence typing. It's a bit of a strange technique in that it gives you something that is a slightly greater resolution of the species, but only if you've got the species right in the first place, which we hadn't. So in a sense, this kind of looks like we've worked backwards, but what we've actually got now is an accurate species identity alongside the lineage, and this can be particularly valid and important when we consider the groupings in the phylogeny. So what we took away from this is that it's in terms of an appropriate workflow for identifying these bacteria. MLST is good for getting a good initial profile, but it should be really used to pull any additional genomes and then classify them based on, say, a core gene technique or something that uses more of the genome data. This then gives you a clear workflow into how to identify the species correctly. But what I found most interesting of all in this data set is that how tightly the industrial isolates group together now in comparison to what we saw previously. As I mentioned, Burkholderia lata can be isolated from a number of environments, including the clinic and essentially environmental samples, so plants, for example. However, with the discovery of the Burkholderia enigmatica type strain, we now see that a lot of these isolates all group together as industrial isolates, and even the Burkholderia enigmatica type strain itself was isolated from hand lotion. So it'll be interesting to see what the unique adaptations are of these isolates that allow them to survive and contaminate industrial products. So this is my acknowledgement slide. There are a lot of people to thank. I'd also like to extend a particular thanks to you, originally just for listening, but also for putting up with technical difficulties. So thank you very much for listening, and I'll take any questions now.
All right, thank you. Thank you, Dr. Cunningham. You have a question from Aya, and she says, great talk, Ed. My question, for the average nucleotide identity method, is that for all nucleotides in the genome or just for the 255 core genes in the other typing method? If for all nucleotides, are there particular regions that have less similarity than the average?
So yeah, that's actually a really interesting question. So the answer to the first part is no. So they're two separate techniques. Now, in terms of what average nucleotide identity compares, and this is one fallibility of the technique, is that it basically compares what it can compare with the quality threshold. So what I mean by that is it will get what it considers a good quality sequence, break it up into, with this particular algorithm I'm looking at, specifically 1,020 base pair fragments of sequences, and it will compare them to each other. So the assumption that you're going in with here is that you have a good quality genome sequence to begin with. But I would say to that as well, that the added value that you get from these type of genomic studies, and this is very much what was done in the Porter publication, is that you can go back to these MLST types and more clearly define the species type once you've actually done that genomics. And now that's sort of been done, people can go back and use the sequence type. So say you haven't got a good quality genome sequence, but you have enough for the multi-locus sequence typing, you've then got sufficient detail to be able to actually identify them down to species level, but it kind of needs genomic studies like this.
All right, let me see if anyone has their hand up. I do have a question. Can you talk more about, so the cost differences between the methods, and when I say cost, not just monetary costs, but like the computing costs, like why someone would lean more towards MLST, like why that would be more enticing when the core genes show to be more accurate or more lead to less misidentifications.
Yeah, of course. So I'm going to answer that in two ways, to be honest, because the first thing I thought about with this technique, and to be honest, when I was doing a lot of this analysis, was cost in terms of outright costs, which wasn't what you were alluding to, but it's a relevant question all the same, and also why you wouldn't use all the information available. So the answer is, of course, if you've got all the information available, use it. But in terms of versatility and also in an industrial setting, it may be more practical for someone to do a scheme that has, say, the amplification of 7 PCR genes, for example. And even some of the genes within the scheme have been shown to be diverse enough to get a decent initial profile, even if it's a partial MST. Some of these databases will actually generate that, will actually give you enough information to try and give you a species identity, even from a limited number of genes. Now, in terms of the actual cost effectiveness, and this is actually a lot of the reason the PhD was funded, it comes down to what the downstream costs are. So it's very tempting to initially say, I'm not going to type this fully with whole genome sequencing because, you know, as we all know, it's a bit more expensive, but it's nothing compared to the cost of a recall. And it's nothing compared to, and that comes down to both the recall of the product, also potential damages to industrial reputation as well. And what we're seeing more and more now is industry are taking this perspective of looking at the downstream costs as opposed to the upstream. So I guess that was where the monetary side of it would come in as well.
Well, thank you. And thank you for your talk.
Yeah.
We're going to move on to our last speaker. Our last speaker is Dr. Innocent Afiki, and he is a professor at the University of Health and Allied Sciences in the Volta region of Ghana. And he's going to talk about neonatal sepsis in a low-income countries teaching hospital. So with that, Dr. Afiki, can you begin?
Thank you and welcome to the last presentation. My name is Innocent Afeke from University of Health and Allied Sciences, Ho, Ghana. Now I'm going to talk to you about neonatal sepsis in a low-income countries teaching hospital. Neonatal sepsis is a major problem in modern medicine. Neonates, especially those born with very low birth weight and also premature, have the risk of morbidity, of morbidity and mortality, and sometimes which will lead to permanent disability. As we could see here, infection causes 18.36% of all the diseases that are diagnosed in the neonatal intensive care. And when we consider those that are born premature, they are also at risk of neonatal sepsis in one way because they use medical devices inserted into the body of these young ones. And also those that are meconium aspiration syndrome, they are also risks of having neonatal sepsis. Currently, coagulase negative are known to be the most cause of neonatal sepsis, although not all the studies that have identified coagulase negative as the most cause of sepsis. Looking at the mortality in the low-income countries and then compared to that of the high-income country, we could see that for Ghana in particular, the rates of infant mortality in 2021 is 32.73 deaths per 1,000 live deaths, compared to that of Germany, which is only 2 deaths per 1,000 live deaths. This shows that neonatal or child mortality is very high in low and middle income countries. So there is the need to determine risk factors that will try to prevent this death in neonates. What is known about the risk factors of neonatal sepsis? For physical factors, most are highly known. We know that infection or sexual infection during pregnancies is a risk factor. We also know that other causes like a fever during pregnancies can be a risk factor. A foul smell, liquor, that is the fluid that came out of a woman after giving birth, if it is smell foul, that is also a risk factor. These are all known in the low-income countries and also in high-income countries. However, high-income countries, they went further to determine molecular characteristics of risk factors of neonatal sepsis. How this is not found in the low-income country. For example, human vaginal microbiota composition has associated preterm birth with risk of neonatal sepsis to vaginal microbiota dysbiosis. Molecular data characterizing the risk factors of neonatal sepsis are largely from the European population and American population. However, data from these areas, geographical areas, cannot be used for low-income countries because it is found out that the vaginal composition of women, Blacks, are different from that of the Europeans, and especially as Ravi and Etar, they have found that women of European ancestry have a community state type of vagina constituting 1, which has a lot of lactobacilli in it, whilst that of African American ancestry, they have the communities state type 4, which lack or have a deficient of lactobacilli. And what are the vaginal microbiota states of indigenous African population? We do not know. However, we know that vaginal microbiota is affected by diet, by lifestyle. It is affected by religion practices and cultural practices. So we do not know the type of community states that are in indigenous Africas. So this study now try to investigate molecular risk factors involved in neonatal sepsis in a hospital in Ghana. So we have 3 objectives. Our first objective is to determine maternal vaginal microbiota as a risk factor for preterm birth with subsequent early onset sepsis. Our second specific objective is to use molecular characterization to determine transmission route or the source of transmission of bacteria that are causing sepsis in this hospital. And our third objective is to use molecular characterization to predict the mechanism of resistant to antibiotics. And we are specifically on antibiotics that are classified as the last resort antibiotics, which is classified by WHO, World Health Organization, Essential Medical Classification Group as the reserve antibiotics. So these are the type of antibiotics which are not used in Ghana or in low-income country. Want to determine how organisms become resistant to that. So this shows the study site. The study was carried out in Ho Teaching Hospital, Ghana. Samples were taken specifically from the neonatal intensive care unit, which is the NICU, and then the baby's ward. These are the type of samples collected from infant and neonatal. Blood culture, mothers, we collected nasal mucosa swab from them and also vaginal swab for microbiota study. We collected nasal swab from clinical staff and students which have not had contact with the hospital, that is first-year students who were admitted into the University of Health and Allied Sciences. And also we took some swabs from objects in the hospital. So this is the study population. As I've stated, mothers, we took 2 samples from their nasal swab and then vaginal swab. For the clinical staff, we took nasal swab from them, and also we administered questionnaire, 3 types of questionnaires for them. First one deal with the type of antibiotic and the type of disinfectant used, and then how often it is used. And then we also administered a standard questionnaire we dealt with burnt out among the clinical staff. For the children, the neonates, we cultured their blood. I must state that there was a limitation here. We should have taken swab from the baby skin or a swab from the nasal, the anal swabs, so that we compare, isolate cultured in their blood with their skin, but we did not do that. Was a limitation of the study. So we took sample from the objects in the hospital, like the weighing scale and the incubators, and also took swab sample from students who have not had contact with this hospital. So I'm now going to talk to you about my first specific objective, which is maternal vaginal microbiota as a risk factor for preterm birth with subsequent early onset sepsis. In this study, we did bacterial cysteine S RNA gene sequencing. We took swabs from mothers. As you could see from the beginning, we had 24 mothers, but when we use inclusive and exclusive criteria, we only had 6 of the samples from the mothers which qualify for the study. We did genomic extraction, and then we performed PCR using trying to amplify the 3V, the variable 3 and the variable 4 variable region of the 16S of the bacteria samples. So this gives the result. Mothers with preterm birth or had a higher microbial diversity. As we could see, these are the two normal women who gave birth, and then those are mothers who gave birth preterm baby, that is spontaneous vaginal preterm birth. So looking at the diversity of the various genus of bacteria, we could see that there is a higher diversity among the preterm group than the normal group. And also we could see that Faecibacterium and Ruminococcus, they predominate in NATE. And all the mothers that we studied, they have a debiosis, that is vaginal debiosis. They lack lactobacilli in their vaginal samples. But an important finding in this study was the detection of Ruminococcus 2 in all the preterm birth mother's vagina, but that was not observed among the control, which were a term birth mother's vagina. So in summary, for this objective, mothers with very preterm birth have higher general diversity of vaginal microbiota than those with term birth. Higher diversity has been associated with unstable pregnancy. And as also genus Ruminococcus, which is also known as Blutia, was detected in the vaginas of mothers who had spontaneous preterm deliveries, but not in those who had term birth, that's control. This genus must be associated with spontaneous delivery. Now I'm going to talk to you about our second objective, specific objectives, which try to use molecular characterization to determine the mode of transmission or source of transmission to this neonate. In this, also samples were taken from mothers' nasal swab, as I stated, and the clinical staff from this environment, and then from the student and blood culture of the babies. These samples were identified using the Maditov, and also were sequenced by Sanger sequencing, which sequenced the TUF gene. This TUF gene was sequenced because studies have shown that it gives a very specificity of coagulase negative, can easily be specified, or a diversity can highly be identified among strains than other 16SR sequencing. So we use 65 Staphylococcus epidermidis and then 35 Staphylococcus hemolyticus for our bioinformatic analysis. We use Staphylococcus hemolyticus and Staphylococcus epidermidis because in our study, these were the common isolates that were cultured from the blood samples of the babies, as you can see, and other studies also support this. So now talk about the maximum likelihood analysis, that the phylogenetic analysis of Staphylococcus epidermidis, and this one's for Staphylococcus hemolyticus. So in this, the results shows that out of the 11 isolates that were cultured from the baby samples, six of the clinical staff matches with this isolate, or they paired with this isolate. So And then when we analyze that of Staphylococcus hemolyticus, we find that out of the 8 isolates that were cultured from babies' blood samples, 5 of the mothers paired with these isolates. So in conclusion or summary of this finding, we could say that clinical staff are more likely to transmit Staphylococcus epidermidis to the babies than the mothers, and the mothers are more likely to transmit Staphylococcus hemolyticus to the babies. Now I'm going to talk to you about our third specific objectives. We deal with antibiotic resistance. And also isolates were cultured from the nasal, from blood samples of baby. Antimicrobial susceptibility was done by the Vitex 2. Whole genome sequences were done for selected samples, that is 15 samples, and antibiotic resistant genes were searched on the website www.card as shown here. So we have identified one antibiotic which falls within the resource or the last resort antibiotics group, which is tingacycline resistant. We found 5 of the Staphylococcus hemolyticus resistant to these antibiotics. So in analyzing their genome genes, antibiotic genes that are found on them, we could see that for those that are resistant to tingacycline, and then we compare them with those that are not resistant to or susceptible to tingacycline. And our data reveals that those that are resistant to tingacycline have more or additional genes than those that were susceptible to tingacycline. So we came out with a network of antibiotics that can be used to predict resistance of Staphylococcus hemolyticus to tingacycline, as shown. So in summary of this objective, molecular mechanism of resistance of Staphylococcus hemolyticus to tingacycline was predicted, and to have consist of network of specific antibiotics gene identified on their genomes. This could be integrated into modern automated techniques to predict resistance to this Staphylococcus species to tingacycline in clinical setting. However, I must say that we need to do further investigation by transforming bacterial cells and trying to take off some of the genes or introduce some of the genes and see if this can be confirmed we can confirm it in the in vitro. So in conclusion of the study, to the best of our knowledge, this is the first time that molecular risk characterization of neonatal sepsis microbiota and bacterial home genomes was done in this hospital, if not in Ghana as a whole. Genomes, our first conclusion that genomes Brominococcus 2 might play a role in spontaneous preterm delivery. Also, vertical transmission of Staphylococcus hemolyticus is more likely than Staphylococcus epidermidis among neonates seen in the neonatal sepsis of this hospital. And then third conclusion, mechanisms of tingacyclines to mechanism of Staphylococcus hemolyticus to tingacycline resistance is predicted in this study. The study came up with some limitations. Sample size is very small. No control database for vaginal microbiota for Ghanaians population is available. Single hospital study needs to be done in multiple hospitals so that it will reflect that of Ghana, and then the duration of the studies was very short. Those are, I would like to acknowledge these people, my professor, my supervisor, Professor Ibrahim Sali, who supervised my work, and also thank Professor Jan Roop for allowing me to do some of the bacteriological tests in his lab, and also thank Professor Ahmed and then for helping me to do some of the bioinformatics. I thank DAD for giving me a scholarship to do this course or to this program in Germany. And also thank the whole teaching hospital for allowing us to take samples in this hospital. With this, I'll say thank you for your attention. And if any question, I'll be ready to answer. Thank you.
All right, thank you, Dr. Afiki. We do have a question in the chat. And Dr. Jefferson Jr. Silva said, very interesting research. I would like to ask if the fungal microbiota will be included in the future as fungi such as Candida are present in this anatomical region.
Yeah, for this, we are doing microbiota. We have another group with my supervisor who are also working on microbiota, which is dealing with fungi. So I think in future, it will be good to include the microbiota in this investigation, vagina investigation, because fungi are also a cause of sepsis. They are also cause of sepsis in neonates.
So, Du Bois, did you have a question? Oh. All right, so if that's all, then thank you, Dr. Afiki, for your presentation.
Thank you, Susan.
Okay, everyone, thank you so much for attending the symposium. This concludes our program for the day. I hope that you enjoyed the research as much as I did, and have a great afternoon. Bye-bye.